There is a field in Tennessee, about an acre in size, surrounded by a chain-link fence topped with barbed wire, where the dead are laid out and studied. Not preserved, not treated, studied in the open air, under the sun and the rain and the heat of a Tennessee summer, in every condition scientists can arrange. Bodies placed flat on soil, face up or face down, bodies submerged in water, bodies locked in the trunks of old cars, bodies buried at varying depths, bodies dressed in clothes and bodies stripped bare, bodies left entirely exposed to whatever comes. The facility is officially called the Anthropological Research Facility at the University of Tennessee, Knoxville. The world calls it the Body Farm. William Bass founded it in 1977. After making an embarrassing mistake that turned out to be one of the more productive errors in the history of forensic science, he was asked to examine the body of a murder victim found in a farm field. He gave investigators a time of death estimate that turned out to be wrong by more than 100 years. The skeleton had been in the ground since the Civil War. Someone had recently disturbed the burial site. Bass had not had the data to know that. Nobody had. There was no systematic research on how human bodies decompose under different conditions, in different soils, in different climates, across different seasons. The question had never been studied with the rigor it deserved. So Bass built a place to gather that data. He convinced the University of Tennessee to give him a small concrete slab and a chain-link enclosure behind the medical center, on ground where the hospital had once burned trash. He and his students began receiving donated bodies and placing them in various conditions, recording everything methodically. When insects arrived, which species arrived first, how the skin changed color and when, how long each stage lasted under what temperature conditions, how burial depth changed the process, how immersion in water altered decomposition relative to surface exposure, how the presence or absence of clothing changed the timeline. Dozens of variables, thousands of observations, year after year. The Body Farm has been running for nearly 50 years now. It has spawned similar facilities in Texas, North Carolina, Colorado, Illinois, Canada, and Australia, because the researchers eventually understood that the decomposition data from Tennessee could not simply be transferred to other climates. The microbes, the insects, the temperatures were all different enough that each region needed its own database. The Body Farm has answered questions that prosecutors and defense attorneys rely on in court. It has trained thousands of forensic investigators, cadaver dog handlers, forensic entomologists, and soil scientists. It has produced a cumulative scientific database that is still being expanded, still being refined, still generating findings that surprise even the people who built it. But somewhere in all that methodical, patient, often confronting science, in the measurements of temperature and insect succession and skin discoloration and the slow collapse of soft tissue, lies a set of facts that most people never sit with long enough to genuinely understand. Your body, after death, does not stop. It transforms. It feeds. It communicates something to the soil and the organisms around it, and the atmosphere itself. The atoms of which you are made do not vanish. They disperse. They move on. They become available again, for other uses, in other forms, across timescales that dwarf any individual life. The question of what happens after we die is one of the oldest questions in human history. Religions have been answering it for as long as there have been religions. Philosophers have been arguing about it for as long as there have been philosophers. Cultures have built entire cosmologies around the journey of the dead. But science has been answering it too, quietly and steadily and with increasing sophistication. In forensic labs and hospital intensive care units and brain imaging centres and EEG monitoring rooms placed next to the beds of dying patients, and the answers science is producing are stranger, more interesting, and more genuinely unsettled than most people know. Tonight, we are going to go through all of it. What happens to the body in the first seconds after the heart stops? What happens in the brain? What the research on near-death experiences actually shows, as opposed to what gets repeated in popularizations. What happens to consciousness, and why that question is significantly less settled than the dominant scientific framework suggests. What happens to the body over days and weeks and years? How those atoms rejoin something larger. And somewhere in the middle of all of this, we are going to sit with the one question science has not yet answered: whether there is something it is like to be in the process of dying, whether experience fades like a light slowly going out, or does something entirely different in those final moments. Take a slow breath. Settle in. This is a long journey. The best way to travel it is quietly. The first thing you need to understand about death is that it is not a moment. This seems like a strange thing to say. We speak of the moment of death as though it is a switch being flipped, a line being crossed, a single event that divides existence from non-existence with clean precision. In legal terms, in medical terms, there is a point at which death is declared. A time is written on a form. A person is pronounced gone. But biologically, this is a simplification. A necessary simplification for practical purposes. For filling out death certificates, for beginning the process of organ donation, for giving families a specific time to anchor their grief. But a simplification nonetheless, one that papers over a process that is far more complex and far more extended than the word moment suggests. Encyclopaedia Britannica's entry on death, in its most recent edition, makes this explicit. The cessation of circulation is a mechanism of death, not a philosophical concept of death in itself. The heart stopping is lethal only if it lasts long enough for critical structures in the brain to die permanently. And those critical structures, as it turns out, are more resilient than they appear, and do not all die at the same rate. Death is a cascade, a sequence of events that unfolds across multiple timescales simultaneously, seconds in some systems, hours in others, days in others still, years in bone. What we call the moment of death is the beginning of that cascade, not its conclusion, not even close. For most of human history, death was defined by the stopping of the heart and the lungs. No pulse, no breath. Dead. This worked adequately as a definition, when those two things reliably meant everything else was going to stop too, and fairly quickly. But medicine intervened. Cardiopulmonary resuscitation was introduced systematically in 1960, following research at Johns Hopkins that combined knowledge of external cardiac massage with artificial respiration. Defibrillators became available in emergency settings through the 1960s and 1970s. Mechanical ventilators could maintain breathing for as long as necessary. Suddenly, hearts that had stopped could sometimes be restarted. Lungs that had ceased to function could be inflated mechanically, while other interventions proceeded. Before 1960, death from cardiac arrest was essentially irreversible by definition. You could not reverse what you could not treat. After 1960, some deaths could be undone. The line that had seemed absolute turned out to be a zone, a period during which the process had begun, but had not yet reached the point of no return. Today, in most countries, legal and medical death is defined by the irreversible cessation of brain activity, specifically the permanent loss of function in the brainstem, the evolutionarily ancient structure at the base of the skull that controls breathing, heart rate, blood pressure, and the arousal mechanisms that make consciousness possible. When brainstem function has permanently ceased, and that cessation has been confirmed as irreversible by clinical testing, a person is declared dead. But even after that declaration, many things continue. Individual cells do not receive the announcement simultaneously. Brain death does not cause every cell in the body to stop at once. Skin cells can remain biologically active for several days. Cells in the cornea can survive for up to 24 hours. Hair and nails do not actually grow after death. That is an old myth, caused by the retraction of dehydrating skin tissue that makes them appear longer. But cells that were doing their work at the moment of death continue on their own diminishing timeline until they exhaust their reserves and are shut down by the spreading consequences of the cascade. There is a hierarchy of death, layered, slow, complex, and most of us, most of the time, do not think about it at all. Let us start at the exact beginning, the moment the heart stops. It can happen at the end of a long illness, the organ weakening progressively for months or years, each contraction a little less forceful than the last. It can happen suddenly, violently, with no warning. A clot dislodging and blocking a coronary artery, an electrical cascade in the conducting tissue transforming the heart's organized rhythm into a useless quiver, a hemorrhage that drops blood pressure below the threshold the heart needs to sustain its own function. It can happen in sleep, in the middle of a dream. However it happens, the effect on the rest of the body is essentially immediate. Within 15 to 20 seconds of the heart's last beat, the brain loses consciousness. This timeline has been documented in studies of pilots subjected to high G-forces in centrifuge experiments, which temporarily drain blood from the brain through gravitational forces, and in patients undergoing certain cardiac procedures where blood flow is briefly interrupted under controlled conditions. The cerebral cortex, the outermost layer of the brain, responsible for thought, language, perception, and the felt sense of being a self in a body in the world, runs entirely on oxygen and glucose delivered continuously by the blood. The brain has essentially no storage for either. It runs on what is being delivered in real time. That delivery stops the moment the heart stops. The cortex's local reserve depletes within 15 to 20 seconds. After that, the metabolic machinery supporting conscious awareness fails. The person loses consciousness. In many cases, there is no experienced transition. Deep, dreamless sleep offers an imperfect but useful analogy. There is no experienced boundary between being awake and the complete absence of awareness that sleep sometimes brings. You are present, and then you are not present, without any experienced interval between those two states. In cardiac arrest, the same thing may happen. The brain stops producing the conditions for experience, and from inside, there is nothing to notice. No experienced darkness. No experienced ending. Simply the absence of the one doing the experiencing. This is the surface of the story. What happens next, beneath the surface, is more complex than almost anyone thinks about when they think about death. Within the first 15 to 20 minutes after death, a window precise enough that forensic investigators use it as a calibration point, the skin begins to pale. Pallor mortis, from the Latin for paleness of death. Blood stops circulating through the capillaries, the smallest blood vessels near the skin's surface. With no pressure driving blood through those tiny channels, they empty. The colouring of living skin, the pinkish or brown or olive or tan tones that come substantially from the blood in those capillaries, drains away. The skin becomes ashen, grey-tinged, regardless of the person's original complexion. Pallor mortis is universal. It happens on the same timeline for everyone, across all skin tones, though it is less immediately visible on darker skin. The body begins to lose heat at the moment of death. Metabolic activity, the constant burning of chemical energy in cells throughout the body, is what produces body temperature. The moment that chemistry stops, the generation of heat stops. The body begins to surrender its warmth to the surrounding environment at a rate determined by the physics of heat exchange. Under typical room conditions, core body temperature drops at roughly 1.5 degrees Fahrenheit per hour. After about 12 hours, the body has reached ambient temperature. This rate is predictable enough to be forensically useful, and it is affected in predictable ways by variables that investigators must account for. Body size relative to surface area, the presence of clothing, air temperature, humidity, whether the body is in sunlight or shade, whether it is immersed in water. All of these have been measured and documented at body farms across multiple climates, giving investigators a quantitative toolkit. Gravity takes over where the heart left off. Blood, no longer being pushed through vessels under active pressure, follows gravity to the lowest points of the body. Wherever the body is resting, the back and buttocks and heels if lying face up, the front and chest if face down, blood begins to pool in the tissues. This pooling creates reddish-blue patches on the skin, called livor mortis, or hypostasis. These patches begin appearing between 20 minutes and a few hours after death, spread over the following hours, and become fixed in place between 8 and 12 hours. The fixing of livor mortis is forensically significant. In the early hours before the blood has fully settled and adhered within small vessels, the patches shift if the body is moved, draining from one area, reappearing in another. After 8 to 12 hours, the blood cells have adhered to vessel walls and the patches are fixed permanently, regardless of repositioning. A body found face down with liver fixation on the back was moved after death. The evidence is in the skin's colour patterns. The body tells this story silently to anyone trained to read it. Muscles relax completely at the moment of death. The jaw may fall open. The hands unclench. The sphincter muscles that maintain continence fail because they require continuous active nerve signals to stay contracted, and those signals stop. Then, between 2 and 6 hours later, those same muscles begin to stiffen. Rigor mortis, the rigidity of death, is caused by one of the more illuminating details of how living muscle tissue works. Muscle contraction and relaxation both require a molecule called adenosine triphosphate, ATP, the cellular energy currency. In a living muscle, chemical bridges form between the protein filaments actin and myosin to produce contraction, then are broken apart by the expenditure of ATP to allow the muscle to relax again. Contraction requires ATP, but so does relaxation. Without ATP, the bridges lock in place. The muscle contracts and cannot release. This stiffening begins in the smallest muscles first, the eyelids, the muscles of the jaw and face, the neck. It progresses through the trunk and into the larger muscles of the limbs. It reaches its peak rigidity at roughly 12 to 24 hours after death. After that, as the muscle proteins themselves begin to be broken down in the early stages of decomposition, the chemical bridges dissolve, and the body returns to limpness over the following day or two. The sequence of onset, peak, and passing of rigor provides another forensic clock. Combined with the state of livor mortis and body temperature, the state of rigor at the time of discovery can narrow the window of time since death considerably. These three markers, pallor, ligor, algor, were among the first systematic tools of forensic pathology, recognized and documented by European physicians in the 19th century. They remain central tools today, now calibrated by decades of body farm research and integrated with newer techniques like forensic entomology and phanotomicrobiome analysis. But while these visible changes are unfolding on the body's surface, something entirely different is happening inside. The digestive system does not receive word when the heart stops. The pancreatic enzymes that break down proteins and fats, the hydrochloric acid produced by the stomach lining, the bile salts stored in the gallbladder. These chemicals do not respond to the cessation of neural signals or to the failure of blood flow. They continue doing their job because their job is a purely chemical process that does not require neural control or active circulation to continue. And their job is to break down organic matter. So they begin breaking down the organs that contain them. This is autolysis, from the Greek for self and dissolution, self-digestion. It begins within minutes of death, though its effects are not visible from outside for several hours. The liver, which is metabolically extremely active and dense with enzymes, shows early autolytic changes. The pancreas, which produces such powerful digestive enzymes that it can begin breaking itself down even in living patients with certain inflammatory conditions. The process known as pancreatitis is particularly vulnerable. The stomach wall begins to be eroded by its own acid. Autolysis is entirely internal in its early stages. From outside, the body may look largely unchanged for the first several hours, but inside, the chemical dissolution has begun. At the same time, the bacteria that have spent the organism's entire lifetime largely confined to the digestive tract start to escape their boundaries. Your gut microbiome, the collective community of microorganisms living primarily in your large intestine, is one of the most complex ecological communities on Earth. A healthy adult gut harbours between 500 to 1,000 distinct species of bacteria, plus archaea, fungi, and viruses, in a total population of trillions of individual organisms. In life, this community is kept in its place by a combination of the immune system, the physical integrity of the intestinal wall, and the chemical environment of the gut itself. The immune system devotes extraordinary resources to maintaining the boundary between the gut microbiome and the rest of the body, preventing the organisms that are welcome in the intestine from entering the bloodstream and organs, where they would cause catastrophic infection. When the immune system fails at death, that boundary collapses. Within hours, bacteria begin crossing the intestinal wall. They enter the mesenteric veins that drain blood from the intestines, travel to the liver through the portal circulation, spread into the systemic bloodstream. They colonize the lymph nodes, the spleen, the other organs. The process is predictable enough in its timing and sequence to be used as a forensic tool. This internal bacterial migration marks the beginning of the phanotomicrobiome. From thanatos, the Greek personification of death, the post-mortem microbial community. And it has become one of the most scientifically productive areas of post-mortem research of the past decade. The thanatomicrobiome proceeds in waves. Different bacterial species colonize different organs at different times, in a succession that reflects both anatomy and microbial metabolic preferences. Researchers have now documented this succession in enough detail to use it as a forensic clock. By analyzing which microbial communities are present in which tissues, in what proportions, investigators can narrow the time of death with increasing accuracy, a new method complementing and extending the classical markers of alga, liga, and liver. Jennifer De Bruyn, a soil scientist at the University of Tennessee, working in close collaboration with the Body Farm, has been documenting not just what happens inside the body, but what happens to the soil beneath it. Her research, published in PLOS One in 2023, quantified the chemical changes in soil directly beneath decomposing human remains. The soil was measurably enriched in nitrogen, phosphorus, carbon, and other nutrients for months and even years after the visible decomposition was complete. The chemical signature of a human life persisting in the earth. Not a metaphor, a measurement. This enrichment is not trivial in ecological terms. Plants growing in that soil take up those nutrients. The organisms that eat those plants carry elements that were once part of a human body into the food web. The decomposing body is not simply ceasing, it is becoming part of what surrounds it in ways that persist and propagate. The gases produced by bacterial activity accumulate inside the body. Bacteria fermenting organic matter produce carbon dioxide, methane, hydrogen sulfide, and a family of volatile organic compounds, including putrescine and cadaverine, named with blunt scientific directness for exactly what produces them. These compounds are responsible for the distinctive odor of decomposition, which forensic investigators universally describe as unmistakable and permanent in memory. Different compounds appear at different stages, and the olfactory composition of decomposition changes over time in ways that trained cadaver dogs can differentiate, and that forensic olfaction researchers are beginning to map systematically. The gases accumulate in the body cavity, causing the abdomen to distend and bloat. This phase, the bloat stage in forensic taphonomy, typically occurs between 2 and 7 days after death in warm conditions, substantially later in cold conditions. The skin, under internal pressure, shows the characteristic changes of active decomposition. Temperature matters profoundly. In the warm Tennessee summer conditions that the Body Farm documented first, a body left exposed on the surface can reach advanced stages of decomposition within a week. The same body in cold winter conditions might show minimal visible change for weeks. These differences are not merely quantitative. Different insect species are active in different seasons. Different bacterial communities dominate in different temperatures. The forensic toolkit has to be calibrated for each region and each season. Water changes everything further. Cold water slows bacterial activity. The absence of aerial insects changes the succession dramatically. The physical effects of immersion differ from those of surface exposure. Bodies in water eventually float when gas accumulation overcomes their negative buoyancy, a timing that has its own predictable pattern that investigators have documented carefully. The insects deserve their own careful account. The arrival of insects at a decomposing body follows biological logic refined over millions of years of evolution. Different species have evolved to exploit different temporal windows in the decomposition sequence, minimizing competition with each other by occupying different ecological niches in what is, from their perspective, an episodically available but extraordinarily rich resource. Blowflies, the caliphorid flies, the green bottles and blue bottles with their iridescent wings are typically the first to arrive, sometimes within minutes of death in warm conditions. The female blowfly detects the volatile compounds released immediately after death, with remarkable sensitivity, following that chemical gradient to its source. She lays her eggs in the natural openings of the body, the nostrils, the corners of the mouth, the corners of the eyes, any wounds present. Under warm conditions, those eggs hatch into first instar larvae within 12 to 24 hours. The larvae develop through 3 instar stages, feeding on decomposing tissue, growing in a timeline that is temperature dependent, and has been measured with sufficient precision that a forensic entomologist examining larvae at a known ambient temperature can calculate the minimum time since death with remarkable accuracy. This is the insect clock, the biological calendar written in the developmental stages of flies. Later in the sequence, other communities arrive. Dermestid beetles, which prefer the drier conditions of later stages when soft tissue is largely gone. Sylphid beetles, which sometimes arrive in earlier stages and predate blowfly larvae. Ants and certain wasps that exploit specific windows. Cheese skippers, hide beetles. Each has its characteristic moment of arrival and departure, its ecological niche in the decomposition timeline. The succession of insect communities is what forensic entomology maps. Before the body farm era, this succession was known in general terms from scattered observations and from work on livestock. The precise, climate-specific, season-specific data that makes forensic entomology legally reliable, defensible in court, subject to cross-examination, came from the body farms. From decades of careful documentation of what arrives, when, under what conditions. What happens when the soft tissue is gone and only bone remains? Bone is a composite material, roughly 60% mineral, primarily calcium hydroxyapatite, and 40% organic, primarily the protein collagen. The organic component degrades relatively quickly under most burial conditions, typically within decades in normal soil. The mineral component is far more resistant under favorable conditions, alkaline soil, dry climate, absence of acidic groundwater. Human bone can persist for centuries, even millennia. This is why archaeologists can reconstruct so much about ancient lives from skeletal remains. The mineral matrix of bone records the chemistry of the food and water the person consumed during life. Stable isotope ratios in tooth enamel indicate where in the world a person spent their childhood. Because different geographic regions have different isotope profiles in their water and food chains, and tooth enamel captures those profiles permanently during the years of formation. Isotope ratios in bone collagen indicate diet over the last decade of life. DNA preserved within bone, protected from degradation by the dense mineral matrix, can, under favorable conditions, be extracted and sequenced thousands of years after death. Even bone, eventually, returns to the soil. Acidic groundwater dissolves calcium hydroxyapatite. Fungal hyphae penetrate bone and break it down mechanically and chemically. The calcium enters the soil solution. Plants take it up through their roots. Animals eat the plants. The calcium in a tree growing near a burial site may have come from bones buried a century before. This is the biogeochemical cycling that has characterized life on Earth for billions of years. Your body, when it decomposes, contributes to all of it. The carbon in your lipids and proteins becomes carbon dioxide and enters the atmosphere, where it may be fixed by photosynthesis into plant tissue within days, or may circulate in the atmosphere for years before being fixed. The nitrogen in your proteins and DNA becomes ammonia, then nitrates, then is taken up by roots. The phosphorus in your bones and cell membranes becomes available in the soil. Every element that made you becomes available again. This is not a metaphor for anything. It is the literal mechanism by which life on Earth sustains itself. Decomposition is not the ending of biological matter. It is the releasing of biological matter back into the cycle. Now let us turn to what was happening in the brain during all of this. Because the brain tells a different and stranger story than any other organ, and it has been telling that story to researchers who were willing to listen for the past decade, with results that have significantly changed how scientists understand the moment of death. Let us go back to the moment of cardiac arrest. The brain loses consciousness within 15 to 20 seconds, as we established. The cerebral cortex dims. The neural processes that sustain the sense of being a self in a body in the world fail, because the metabolic fuel that sustains them is exhausted. The person, from the inside, is simply gone. But in at least some cases, the brain does not simply go quiet. In 2013, Jimo Borjigin, a neuroscientist at the University of Michigan studying the pineal gland and circadian rhythms, was running an experiment on rats. She induced cardiac arrest while monitoring brain electrical activity with electroencephalography, expecting to see declining activity as the brain lost oxygen and began to shut down. Instead, in the 30 seconds after cardiac arrest, she saw a surge, not a continuation of prior activity, not the last flickering of a failing system. A new onset of high-frequency gamma oscillations, electrical activity in the 30 to 80 Hz range, that was stronger and more coherent than what the rats had shown during normal waking life. Gamma waves in living brains are associated with attention, complex cognition, working memory, and the integration of information across different brain regions. They are the neural signature of high-level conscious processing. These rats, dead, were showing them with greater amplitude than they had shown in life. Batthyány published the finding in 2013. It was received with a mixture of fascination and skepticism. Rats cannot report their experiences. The leap from gamma activity to conscious experience, while suggestive, remained speculative. The field noted the finding and moved on. Then, in 2022, a case appeared in the human literature. An 87-year-old patient at the University of Louisville was being monitored by EEG for seizure activity. During the monitoring, the patient died of a heart attack. The EEG was running continuously, before, during, and after cardiac arrest. The neurosurgeon Ajmal Zamar, examining the data afterward, found exactly what Batthyány had found in rats. A surge of gamma oscillations in the period surrounding death, appearing specifically in the temporal and parietal regions of the brain, the temporal-parietal junction, the region associated with the integration of information from different sensory modalities, the region that, when electrically stimulated in epilepsy patients during surgical procedures, sometimes produces the sensation of leaving the body, the region whose disruption causes disturbances in the sense of being a self located in a particular position in space. Zamar noted that the pattern of gamma activity matched what is seen in living brains during memory recall, during dreaming, and during meditative states. He stated explicitly that this alignment with near-death experience accounts was striking. One case is one case, but the finding was real. Then 2023 arrived and the evidence base shifted. The May 2023 paper from Batthyány's team at the University of Michigan, published in the Proceedings of the National Academy of Sciences, was the first systematic study of gamma oscillations in dying human patients. 4 patients were monitored continuously with EEG throughout the dying process, all comatose, all beyond medical intervention, all with family consent for life support removal. 2 of the 4 showed the surge. In the seconds after ventilator removal, as their hearts stopped, their brains showed a dramatic increase in high-frequency gamma oscillations, appearing specifically in the temporal parietal junction, stronger than any activity they had shown in the hours before death. These 2 patients showed, at the moment of death, a neural signature that in a living brain would indicate complex, coordinated conscious processing. Sam Parnia, commenting on the study for the journal Science, called it evidence of a marker of lucid consciousness in the dying brain. He went further. We have this binary concept of life and death that is ancient and outdated. The research was showing something that neither life nor death, in their conventional meanings, adequately described. The same year, Parnia published the results of the largest study of consciousness during cardiac arrest ever attempted, the AWARE-2 study. Conducted across 25 hospitals in the United States and United Kingdom between 2017 and 2020, 567 patients who suffered in hospital cardiac arrest, a subset of 85 with continuous brain monitoring during resuscitation. Fewer than 10% of the 567 patients recovered sufficiently to be discharged. Of those who survived and were interviewed, a significant proportion reported recalled experiences from the period of cardiac arrest that Parnia's team concluded were qualitatively distinct from hallucinations, delusions, dreams, or CPR-induced confusion. The experiences described were structured and consistent. Patients described separating from their bodies and observing the resuscitation from a vantage point above or outside their own physical position. They described this with specificity, the positions of staff in the room, the equipment being used, details that were subsequently verified against medical records. They described encountering deceased relatives. They described a panoramic review of their lives, experienced not as linear memory, but as simultaneous re-experience seen from multiple angles, including the perspectives of the people they had affected. They described profound peace. They described arriving at a threshold or boundary. Parnia's team noted that these experiences were characterized by a quality of lucidity, of heightened, expanded awareness that was the opposite of the confusion and fragmentation that characterizes ordinary delirium or hypoxic brain states. The EEG data from the monitored subset reinforced the University of Michigan findings. Even up to an hour into resuscitation, In patients who were clinically dead, the brain showed intermittent spikes of electrical activity, including gamma, delta, theta, alpha, and beta waves, the full spectrum of waking brain activity appearing in brains that should, by conventional understanding, have been producing nothing at all. Even up to an hour into the resuscitation, Batthyány said, we saw spikes, the emergence of brain electrical activity, the same as I have when talking or deeply concentrating, in flatlined brains. Bruce Greyson, the veteran near-death experience researcher at the University of Virginia, offered a methodological caveat. The patients who showed gamma activity and the patients who reported near-death experiences were not the same patients. The direct correlation was not established. This is a legitimate scientific point. The data as it stands does not demonstrate that the gamma activity caused or accompanied the experiences. But the data does establish something important. Dying brains, in at least some cases, show the neural signature of complex conscious processing at the moment of death. And cardiac arrest survivors, in statistically meaningful numbers, report structured experiences from that period that are qualitatively distinct from ordinary altered states. Whether those two facts are directly connected and what they mean together is genuinely open. But the openness is scientific, not philosophical. It is the openness of a field that is gathering data, not the openness of permanent ignorance. Among the most consistent elements in near-death experience accounts, across cultures, across centuries, across the accounts of people who have had no prior knowledge of the phenomenon, is the life review, not memory in the ordinary sense, not a linear replay of events from the beginning to the end. Something described as simultaneous, all of one's life present at once, accessible from any direction, felt rather than simply recalled. Survivors describe re-experiencing significant events from the perspective of the other person involved, feeling what that other person felt, understanding in an experiential rather than intellectual way what their own actions meant to the people around them. Many describe it as including a quality of moral evaluation that is neither condemning nor forgiving, but simply complete and clear, as if every action and its consequences were simultaneously present without the distortions of memory or self-interest. The scale and vividness described is unlike ordinary memory. People report experiencing vast amounts of their lives in what, by external measurement, was a very brief period. They describe time as not linear, as having dissolved its usual sequential structure. Some describe the experience as the most significant and real thing that has ever happened to them. Ajmal Zamar, examining the EEG data from the dying 87-year-old patient in 2022, noted that the gamma activity was highest in the temporal parietal junction, the region associated with autobiographical memory retrieval, with the integration of memories from different sensory modalities, and with the felt sense of self in time, the same region that, in living brains, is most active during the retrieval and reliving of personal memories. The neural signature and the phenomenological accounts were pointing at the same place. Whether the activity Zemmar measured was the biological substrate of what survivors describe, or a coincidence, is not something a single case study can determine, but it is a coincidence that serious researchers are now investigating with the tools available to them, and it is a coincidence that grows more significant with each study that adds a similar data point. Now I want to turn to a phenomenon that cuts even deeper into these questions, one that has been almost entirely absent from public discourse until very recently, and one that I think is among the most scientifically important anomalies in contemporary neuroscience. Terminal lucidity. The phenomenon is old. Physicians observed and documented it in medical literature throughout the 19th century. William Monk, writing in his 1887 medical text on end-of-life care, described patients who had been cognitively absent or unresponsive for extended periods, weeks, months, recovering unexpected clarity in the hours or days before their deaths, speaking coherently to family members they had not recognized in years, recounting specific memories from decades past with apparent accuracy, expressing emotions with precision and intention, saying, in effect, goodbye, and then dying shortly afterward. The pattern was noted and recorded, and then largely filed under the category of things that happen but cannot be explained, and therefore need not be pursued. Alexander Batthyány, director of the Viktor Frankl Institute in Vienna, and a research professor at Pázmány Péter University in Budapest, decided in the early 2000s that it deserved systematic study. He began collecting documented cases, working with healthcare professionals, families, and institutional caregivers to gather detailed accounts of episodes, what the person said, how long the episode lasted, what their condition had been before, what happened after. In collaboration with Bruce Greyson, who had spent decades studying near-death experiences at the University of Virginia, Batthyány published the first systematic analysis of what they called paradoxical lucidity, the preferred scientific term, intended to emphasize the paradox relative to the underlying neurological condition. The findings were striking in their clarity. In 38 documented cases of paradoxical lucidity in patients with severe cognitive impairment, primarily advanced Alzheimer's disease, but also patients with profound brain damage from tumours, strokes or prolonged coma, 44% of the lucid episodes occurred within 1 day before the patient's death. 31% occurred within 2 to 3 days. Only 6% occurred more than a week before death. This clustering is not random, it is a pattern, a strong, consistent pattern that argues for a biological relationship between proximity to death and the temporary return of cognitive clarity. The episodes themselves were substantial in duration. 3% lasted less than 10 minutes. 24% lasted between 30 minutes and an hour. 29% lasted several hours. 11% lasted an entire day. And the patients experiencing these episodes were not patients who had been showing gradual improvement. They were patients in late-stage Alzheimer's disease, patients whose brain tissue had been so devastated by amyloid plaques and neurofibrillary tangles that they could not recognize their own children, could not name common household objects, could not form grammatical sentences. Patients who had been institutionalized for months or years with no meaningful cognitive contact with the outside world. They were, briefly, fully themselves, recognizing family members by name, recounting specific memories from decades in the past with apparent accuracy, memories their families could verify, expressing nuanced and appropriate emotions, conducting conversations that were not just coherent but fully present, engaged, and aware, and then dying. The neurological problem this creates is severe. The standard framework of cognitive neuroscience is explicit. Cognitive function is dependent on the integrity of the neural tissue that supports it. If neurons are dead or damaged beyond function, the corresponding cognitive functions are lost. This is not just theory. It is supported by an enormous body of clinical evidence. Specific lesions produce specific, predictable deficits. Restoration of function in areas of permanent neurological damage does not occur by the mechanics of the framework. There is no proposed mechanism by which a patient with the degree of neural destruction characteristic of late-stage Alzheimer's disease could suddenly and temporarily recover cognitive functions that require the very neural architecture that has been destroyed. George Mashaw at the University of Michigan, one of the neuroscientists who led the 2023 gamma wave study, engaged directly with this puzzle in a 2019 paper in Alzheimer's and Dementia. He concluded that paradoxical lucidity, if systematically confirmed, challenges current assumptions and highlights the possibility of network-level return of cognitive function in cases of severe dementias. He called for systematic investigation using modern tools, prospective EEG monitoring, brain imaging during lucid episodes, standardized case collection across large populations. That investigation is now underway. The National Institute on Aging has funded research specifically on paradoxical lucidity. Sam Parnia's group at NYU Langone is involved in prospective studies placing EEG monitors on terminally ill patients with dementia, aiming to capture the neurological signature of lucid episodes when they occur. A Dutch survey found that 7 out of 10 nursing home caregivers had observed such episodes in their patients at least once in the preceding 5 years. 7 out of 10. Among the people who work most closely with the dying, this is not a rare anomaly. It is a regular observation. It has been regular for as long as people have been caring for the dying. It has simply never been systematically studied. I want to take a clear position here, because the topic requires it. The dismissal of terminal lucidity as wishful thinking as grieving families misinterpreting any small sign of responsiveness in a beloved person at the end of their life, does not account for the systematic pattern Batthyány documented. It does not account for the frequency of professional caregiver observations in populations with no personal attachment to the patients. It does not account for the best documented cases, which involve nursing staff and physicians observing patients they have never met before, documenting what they see in medical records with professional specificity. Steven Pinker dismissed this research publicly in the Wall Street Journal, calling it wishful misinterpretation, suggesting that when there is desperation to communicate with a loved one, any glimmer of responsiveness can be exaggerated. Batthyány replied directly in the same publication, noting that serious anomalies demand serious inquiry, not reflexive rejection. He specifically pointed out that Pinker's explanation requires systematic distortion by professional caregivers with no personal stake in the outcome, which the data does not support. The appropriate scientific response to an anomaly that does not fit the existing framework is investigation, not assumption that the anomaly cannot exist because the framework says it cannot. The history of science is full of anomalies that turned out to be real and that required framework revision. The appropriate posture toward paradoxical lucidity is the posture Mashore and the National Institute on Aging have adopted: serious, funded, prospective, rigorous investigation. The mechanism is unknown. The phenomenon is documented. Those are the two facts the science requires holding simultaneously, without collapsing the uncertainty in either direction. Let me turn now to one of the most surprising findings in recent death research, one that emerged entirely by accident and that has implications that reach beyond the forensic and into the deepest questions about what biology actually is. The zombie genes. In 2016, a team led by microbiologist Peter Noble at the University of Washington was running what should have been a routine control experiment. They were testing a technique for measuring gene expression, the process by which information in DNA is read and translated into proteins, and they were using tissues from recently dead zebrafish as a baseline, expecting to see declining activity as cellular processes wound down after death. They did not get what they expected. After the zebrafish died, roughly 1% of their genes showed not declining activity, but increasing activity. Genes that had not been significantly active in the living organism were switching on after death. This seemed so implausible that the team assumed they had made a technical error. They checked their instruments. They ran the experiment again. Same result. They switched to mice, euthanized under controlled conditions. Same result. What they were documenting was real. After an organism dies, a specific subset of genes, not random genes, not all genes, but a particular and apparently non-random selection, activates. More than 1,000 genes in total across the two species. In mice, 515 genes showed post-mortem activation, most within the first 24 hours. In zebrafish, 548 genes, with some remaining active up to 4 days after death. In human tissue, brain samples, blood samples, skin samples, researchers at Barcelona's Centre for Genomic Regulation subsequently confirmed the phenomenon, finding comparable patterns of post-mortem gene expression in human cadaver tissue. When Noble and his team categorized these zombie genes by function, they found consistent themes. Genes associated with immune and inflammatory response, as if the cellular machinery were responding to catastrophic infection or injury. Genes associated with stress response. Genes associated with cancer growth promotion. And the most striking category, genes associated with embryonic development. Genes that are highly active in rapidly dividing embryonic tissue, in the early days of building an organism from scratch, were activating in dead tissue. The most coherent explanation Noble proposed was regulatory. In a living organism, these developmental and emergency response genes are suppressed by regulatory networks, by other genes and molecular systems that maintain the adult organism's stable state and prevent inappropriate developmental signaling. When the organism dies, the active maintenance of those suppressive networks fails. The brakes come off. The underlying programs, which were always present but held down, briefly run. The dead body, on this interpretation, is in a state that biochemically resembles the early embryo, not because it is growing, but because the same molecular toolkit is running in the absence of the regulatory controls that normally distinguish between development and maintenance. Something in the cellular logic, finding itself suddenly without guidance, reverts to its most fundamental default programming. Noble himself described the finding as the most bizarre thing I have ever seen. He also articulated its broader implication. The headline of this study is that we can probably get a lot of information about life by studying death. The regulatory networks that maintain biological stability become visible when they fail. The practical implication his team identified involves organ transplantation. Recipients of donor organs have substantially elevated cancer risk for certain malignancies. Non-Hodgkin lymphoma, kidney cancer, liver cancer among them. If cancer-promoting genes are activating in donor tissue during the period between death and transplantation, those activated states may persist in the transplanted organ and contribute to cancer risk in the recipient. Understanding the phanetotranscriptome, the full gene expression profile of post-mortem tissue, could eventually inform interventions that reduce this risk, potentially saving lives downstream from the donation itself. Each major organ has its own clock after death, and the differences between organs reflect millions of years of evolutionary history. The heart and lungs are the most oxygen-sensitive organs. They can remain viable for transplantation for only 4 to 6 hours after death, even under optimal conditions of cold preservation. This narrow window reflects the extreme metabolic demands of cardiac and pulmonary tissue. Cells that have evolved to be perpetually active, perpetually supplied, with essentially no adaptation for periods of deprivation. The liver is more resilient. 8 to 12 hours under cold preservation. The liver's metabolic versatility, the range of chemical processes it manages, the diversity of its cellular functions, gives it some capacity to tolerate ischemic stress before reaching irreversible damage. The kidneys are the most resilient major organs, 24 to 36 hours. Kidney cells, which evolved to regulate blood volume and pressure in environments of variable hydration, have developed cellular adaptations that allow them to tolerate brief periods of reduced oxygen delivery better than heart or lung cells. A single organ donor can provide up to 8 organs: 2 lungs, a heart, a liver, 2 kidneys, a pancreas, intestines. One person's death can provide life, years or decades of functioning life, to 8 different people. The surgery to recover those organs takes between 2 and 4 hours, conducted with the same careful sterile technique and the same level of respect as any living patient's operation. What happens in the donor body after the organs are removed is not fundamentally different from what happens in any other body. The same cascade of autolysis and bacterial migration and decomposition follows. The biology does not distinguish, but the donated organs continue. The heart keeps beating in another chest, pumping blood through vessels in a body that would have died without it. The kidneys keep filtering in another body, hour after hour, year after year. The liver performs the hundreds of metabolic tasks it was built to perform, in a different organism, with a different genetic identity, but doing its work the same way it would have in the donor. This is one of the more extraordinary things medicine has made possible. The literal biological continuation of parts of one person inside the living body of another, not metaphorically, biochemically, concretely. The cells of the donor, incorporated into the recipient, continue performing the functions they were shaped by evolution to perform, for years or decades. The atoms deserve their own careful account, because I think it is the most overlooked aspect of what death actually is, and what life was. Your body, at this moment, contains approximately 7 octillion atoms. Written as a number, that is a 7 followed by 27 zeros. The number is too large to carry any intuitive weight, but what matters is what those atoms are and where they came from and what will happen to them. Your body is mostly water by mass, about 60%. Water is 2 parts hydrogen to 1 part oxygen. So oxygen is your most abundant element by mass, hydrogen third. Carbon is second, the scaffolding atom of all organic chemistry, the element on which biological complexity is built, the atom that makes the molecules of life possible. Then nitrogen, essential for every protein and every strand of DNA. Then calcium, mostly in your bones and teeth. Phosphorus in bones and in the backbone of every DNA molecule you carry. Sulfur, iron, potassium, sodium, magnesium, more than 24 trace elements in smaller amounts. None of these atoms were made in your body. They were assembled into you from the food you ate, the water you drank, the air you breathed across the entire span of your life. Before they were in your food, they were in the organisms your food came from. The plants and animals and fungi and bacteria that fed into the systems sustaining you. Before that, they were in the soil and the ocean and the atmosphere. And before that, in the case of every element heavier than hydrogen and helium, they were forged in the nuclear fusion reactions of stars that lived and died long before our solar system existed. In stellar interiors, where temperatures reach tens of millions of degrees and the gravitational pressure is beyond any terrestrial analogy, hydrogen fuses into helium. Helium fuses into carbon. Carbon into oxygen. Successive cycles of fusion in the largest stars produce progressively heavier elements. When a massive star exhausts its fusion fuel and collapses in a supernova explosion, those elements scatter across interstellar space in an expanding shell of gas and dust that may eventually become the raw material for new planetary systems. The cloud of gas and dust that condensed to form our solar system, roughly 4.5 billion years ago, contained atoms forged in stars that lived and died before our sun was born. Some of those atoms, over billions of years, through the chemistry of a forming planet and the emergence of life and the evolution of biology and the long chains of ecological succession, ended up in you. Your carbon atoms have been through stars, more than one. This is not a metaphor or a spiritual consolation. It is astrophysics. It is what the isotope ratios in ancient materials tell us about the nuclear history of the matter that makes up the solar system. When you die and your body decomposes, those atoms return to circulation. The carbon in your lipids and proteins becomes carbon dioxide, exhaled by the bacteria consuming your tissue, released into the atmosphere. Plants fix that carbon into glucose through photosynthesis, sometimes within days of its release. Animals eat the plants. The carbon is in the food web again, moving through organisms within weeks or months. Within a year, atoms that were part of your nervous system may be in the leaves of a tree nearby, in the body of a bird, dissolved in a stream. The nitrogen in your proteins and DNA is converted by bacteria into ammonia through ammonification, then converted into nitrates through nitrification, then absorbed by plant roots and built into new proteins. The phosphorus in your bones and cell membranes becomes available in the soil solution and enters the local nutrient cycle. The calcium in your bones persists longest, but eventually it too dissolves and circulates. De Bruyn's 2023 research at Tennessee measured this directly. The soil beneath decomposing human remains showed elevated nitrogen, phosphorus, and carbon for months and years after the visible decomposition was complete. The chemical enrichment was real and measurable, not a metaphor, not a consolation offered to the bereaved. A soil chemistry measurement published in a peer-reviewed journal. This is what it means, in the most literal and unsentimental sense, to return to the earth. The atoms return. They become part of other living things. The imagery that religion and poetry have used for millennia turns out to be, at the level of molecular chemistry, chemically precise. Now I want to stay with the consciousness question, because I believe it deserves more serious and more honest attention than it typically receives. The standard materialist position on consciousness and death is clear. Consciousness is a product of neural activity, and when neural activity permanently ceases, consciousness ceases with it. End of story. This position is held by many thoughtful, rigorous scientists, and it is motivated by an enormous body of evidence. Specific brain lesions produce specific predictable deficits in specific aspects of conscious experience. Damage to the visual cortex impairs vision in predictable ways. Damage to Broca's area impairs speech production in predictable ways. Damage to the hippocampus impairs the formation of new memories in predictable ways. The correlation between brain state and conscious state, across an enormous range of observations in neurology and cognitive science, is among the most robust findings in all of science. I am not dismissing that evidence. It is real. But two problems remain unresolved within that framework, and they are not peripheral. The first is what philosopher David Chalmers named the hard problem of consciousness in 1995. The easy problems of consciousness, explaining how the brain integrates information, directs attention, produces coordinated behavior, are tractable in principle because they involve functional relationships between neural processes that can be studied with standard scientific methods. The hard problem is different. It asks why any of this neural processing is accompanied by subjective experience at all. Why is there something it is like to see the color blue, to feel physical pain, to hear a piece of music as beautiful? Why is brain processing not simply information processing with no felt quality, no inner dimension, no subjective perspective on itself? No theory of consciousness has answered this. After 3 decades of intense neuroscientific investigation, the relationship between neural activity and subjective experience remains unexplained. The neural correlates of consciousness, the specific patterns of brain activity associated with conscious states, have been studied extensively, but no one can explain why those patterns are accompanied by experience rather than simply by more neural processing. Christof Koch, who made a public bet in 1998 with Chalmers that the neural correlates of consciousness would be identified by 2023, conceded publicly in 2023 at a conference in New York, presenting Chalmers with a bottle of 1978 Madeira on stage. The neural correlates had not been found. The hard problem remained hard. The failure was honest and significant. The second problem is the data we have been examining. If consciousness simply is neural activity, and if neural activity ceases at death, then the gamma surges in dying brains are informative noise, the random electrical discharge of failing tissue. This is possible. There is no way yet to prove it is not. But if it is noise, why does it appear specifically in the temporal parietal junction, the region most associated with out-of-body experience and autobiographical memory? Why does it appear with the coherence and organization of gamma activity associated with complex conscious processing in living brains? Why does it appear at the moment of death in 2 of 4 patients who were studied, not continuously throughout the dying process, suggesting it is event-related rather than random background activity? These coincidences do not prove experience, but they make the noise interpretation less comfortable than it might initially appear. Christof Koch, writing as a commentary on the 2023 Gamma Wave paper in PNAS, titled his piece with Dylan Thomas, "Do Not Go Gently Into That Good Night." He noted that the dying brain appears to be doing something unexpected and complex, and that science has barely begun to understand it. In April 2026, Koch presented at the Bial Foundation Symposium in Porto the argument that consciousness may not be something the brain creates, but something more fundamental to the structure of reality that neural systems instantiate or access. This is a version of panpsychism, the philosophical view that consciousness, or something relevantly similar to it, is a basic feature of physical reality rather than an emergent product of sufficiently complex biological information processing. Panpsychism is not a fringe position. It is taken seriously by a significant and growing minority of philosophers of mind, and a smaller but non-trivial number of consciousness researchers. It is not the same as claiming consciousness survives death. A panpsychist view is compatible with the dissolution of a particular pattern of consciousness at death. But it does unsettle the assumption that death is simply the end of experience, because it raises the possibility that experience has a different and more fundamental relationship to physical reality than the simplest materialist account assumes. I am not claiming consciousness survives death. I do not know whether it does. What I am claiming is that the question is scientifically open in a way it was not 30 years ago, and that the confidence with which some scientists dismiss the question reflects commitment to a framework, not analysis of the evidence. Let me bring everything together now, because I think the full picture deserves to be seen as a whole. Death is a process that unfolds across multiple timescales simultaneously. The heart stops. The brain loses consciousness within seconds. The body cools, the blood pools, the muscles stiffen and then relax again across the following hours. Bacteria migrate from the gut and begin the biological process of dissolution. Digestive enzymes self-digest the organs that house them. Insects arrive in an ecologically calibrated succession. Gases accumulate. Soft tissue breaks down over days and weeks. Bone persists for decades or centuries before it too returns to the soil. Through all of this, individual cells continue their own timelines. Genes activate in dead tissue, specific genes, in non-random patterns, doing something our categories of life and death were not built to describe. The thanatomicrobiome proceeds through its succession, a living ecosystem carrying out biological work inside a dead body. At the moment of death, in at least some cases, the brain surges. Gamma oscillations appear in the temporal parietal junction. The neural signature of complex, coordinated conscious processing appears in tissue that has no heartbeat, no blood flow, no oxygen delivery. 2 patients in the 2023 University of Michigan study, 1 patient in the 2022 Louisville study, multiple patients with brainwave activity during resuscitation in the AWARE-2 data. Whether this activity accompanies experience is unknown. The question is being investigated with increasing rigor. In some patients with severe, apparently irreversible neurological damage, there are documented episodes of restored cognitive clarity in the days before death. The mechanism is unknown. The pattern is robust. 7 out of 10 nursing home caregivers have observed it. The National Institute on Aging is studying it now, with tools that did not exist a decade ago. The atoms return to circulation. The carbon, nitrogen, phosphorus, calcium of your body will be in the atmosphere and in soil and in other organisms within months or years of your death. The process has been measured. The enrichment has been quantified. The chemical signatures persist in the earth. The question that remains genuinely open, not philosophically open, but scientifically open, is whether the self that was assembled from those atoms experiences the dissolution, whether there is something it is like to die, whether the gamma surge is felt, whether the life review is experienced from the inside with the quality that survivors describe. The hard problem of consciousness means this question may not be fully answerable with current tools, because the relationship between neural activity and subjective experience is not understood, even in normal waking life. The dying brain research has made it an empirical question rather than a purely philosophical one. But the empirical methods are still limited, still indirect, still dependent on inferences from external measurements to internal states. The scientists who are asking these questions most carefully, Batthyány, Borjigin, Zemar, Batthyány, Mashor, Koch, are doing so with genuine intellectual honesty. They are reporting what the data shows, acknowledging what it does not show, and continuing to gather more. This is what science does when it is doing its best work. There is one more thing worth saying about what this field of research does to the people who work in it. Batthyány has spoken publicly about how studying near-death experiences has changed his understanding of death, and that the change has not been toward despair. The dying brain's final activity, in the data he has gathered, does not look like simple shutdown. It looks like something more active, more ordered, more interesting than a machine turning off. Batthyány, studying paradoxical lucidity, has said that whatever its eventual explanation, the phenomenon suggests that something persists in the person even when the brain appears to have lost the capacity to express it. Whether that something persists beyond death, the data does not say. But it suggests that the model of the relationship between brain and person is less complete than the consensus had assumed. Koch, after conceding his bet with Chalmers, continued his research. His 2024 book describes consciousness as potentially among the most fundamental features of reality, not a late biological invention, but something that the universe has been doing in various forms since long before there were brains to do it. Whether this changes anything about what death means for consciousness depends on what consciousness actually is, and that, he acknowledges, remains unknown. The humility these researchers display is appropriate. Death is the one phenomenon that every person will experience, and that no person can reliably report back from. The window it offers into the nature of consciousness and existence is the most restricted window in science. The data that comes through it is fragmentary, contested, difficult to interpret. But it is data, real data, gathered carefully, analysed rigorously, contested seriously. And it is pointing somewhere, even if we cannot yet see clearly where. Let the science settle in you now, in the quiet of this hour. The body cooling and stiffening, the bacteria spreading, the insect succession beginning, the atoms beginning their slow return to the cycles that sustain all life. The brain surging, briefly, in some deaths, in the regions most associated with memory and self and awareness. The genes activating one last time in dead tissue, doing something no existing word quite captures. the microbiome that was yours, dispersing into the soil and the atmosphere, carrying chemical signatures of the life it had been part of. The atoms older than the Earth, beginning their next iteration. You do not need to resolve what it means. Some questions are designed to travel with a person through their life, rather than give them somewhere to stop and put the question down. They are companionable questions. They keep you honest about how much you know and how much you do not. What the science has given tonight is an honest account of a process that is stranger, more active, more biologically complex, and more genuinely mysterious than the simple binary of alive and dead suggests. The process is not an ending. It is a transformation of enormous complexity, some of it documented and understood, some of it newly discovered and still being interpreted, some of it still barely glimpsed at the edges of what current instruments can measure. Some of what happens after death is well established, some is newly found, some is unknown, including the thing that matters most to every person who will die. Whether experience persists in any form beyond the last heartbeat. The instruments are still measuring, the studies are still running, the EEG monitors are being placed in more hospital rooms, the body farms are accumulating more data, the phanotomicrobiome is being sequenced in finer resolution. The consciousness researchers are still searching for the answer that Christof Koch bet 25 years of his career he would find. They're still searching. The night holds them alongside you. Sleep is, perhaps, the closest thing available to practice for the letting go. And if science is right that something in us surges at the end, if those gamma waves carry anything at all, then the boundary between sleep and death may be, as the poets and philosophers always suspected, less absolute than the machines suggest. Let that be something to rest with, rather than something to worry about. The questions are good company. There is something else the science has found that most people never hear about, and it concerns the question of how we think about death itself, not just what happens during it. The definition of death has a history, and that history reveals that what we call death has never been as clear or as fixed as the word implies. The ancient world defined death simply: the heart stopped, the breathing stopped, the person was gone. In a world without CPR, without defibrillators, without intensive care units, this was accurate enough to be practical. If the heart stopped, it stayed stopped. If breathing ceased, it did not resume. The definition and the reality coincided. The first cracks appeared long before modern medicine, however. Physicians in the 18th century began to notice, with growing alarm, that some people declared dead were not dead. Premature burial became a subject of serious anxiety in Europe, not because of supernatural fear, but because of documented cases of people who had been placed in coffins or buried while in states that mimicked death so completely that the medical tools of the era could not distinguish them from genuine death. Catalepsy, severe hypothermia, certain poisonings, certain cardiac arrhythmias, all could produce states indistinguishable from death to an observer with only touch, sight, and hearing to work with. The London Humane Society, founded in 1774, became one of the first institutions dedicated to resuscitation, initially of drowning victims, but then more broadly. By 1796, they claimed to have resuscitated over 2,000 people who had been presumed dead. This was not a small number. The society understood, a full century and a half before the invention of modern CPR, that what the era called death was sometimes a reversible state. The development of the stethoscope in the early 19th century helped. Physicians could now listen for heart sounds that were too faint to detect otherwise. But the fundamental conceptual problem remained. The definition of death was based on observation of cessation, and cessation of heartbeat and breathing, it turned out, was not always permanent. The 20th century brought the problem to a crisis point. The development of cardiac surgery, of the heart-lung bypass machine, of mechanical ventilators, of cardiopulmonary resuscitation, of defibrillation, these technologies revealed that what had been considered the irreversible end could sometimes be reversed. Hearts stopped during surgery and were restarted. Lungs kept still for hours during procedures resumed their function. People who had no heartbeat and no breathing for minutes were resuscitated and made full recoveries. The medical and legal establishment needed a new definition of death that tracked irreversibility rather than mere cessation. The solution was brain death. In 1968, a committee at Harvard Medical School published a report proposing criteria for irreversible coma as a new definition of death. The criteria included unresponsiveness to external stimuli, absence of spontaneous movement, absence of reflexes, absence of spontaneous breathing, and a flat electroencephalogram. No detectable brain electrical activity. These criteria, refined and formalized over subsequent decades, became the basis for brain death determinations that are now standard in medicine and law in most countries. The significance of this shift is considerable. For most of human history, death was defined by the stopping of the body's most visible and essential activities, heartbeat and breathing. From the late 20th century onward, death has been defined by the permanent cessation of a particular organ's function, the brain, and specifically the brain stem. The heart can stop, and a person can still be alive, as long as the brain continues. The lungs can cease, and a person can still be alive, as long as the brain is functioning. What defines the person is now understood to be the brain. This redefinition reflects a genuine philosophical insight. The heart is a pump. A remarkable, complex, exquisitely evolved pump, but ultimately replaceable, and in fact, replaced by transplant thousands of times every year. The brainstem, by contrast, is not replaceable. It is not merely an organ that performs a function. It is the structure that integrates the functions of all other organs, that maintains the internal environment in which consciousness is possible, that regulates the autonomic processes that keep the body viable. Without it, nothing else runs sustainably. But this definition, too, has its complications. Brain death requires that all brain activity has permanently ceased. Yet, as we have seen, the brains of some dying patients surge with gamma activity at the moment of death. Does this activity count as brain activity? Is the brain of a patient showing post-mortem gamma oscillations still alive, by some definition? These are not merely philosophical questions. They have practical implications for the timing of organ donation, for the interpretation of EEG readings, for the legal determination of death. Batthyány himself has said that the binary concept of life and death needs to be replaced by something more nuanced. A recognition that death is a process, and that different components of the organism reach irreversibility at different times, and that the overall determination of death requires integrating information about multiple systems, rather than finding a single threshold moment. This is not a comfortable conclusion for a legal system that requires a specific time to write on a death certificate. But it is the conclusion the biology supports. The microbiome we carry through life shapes us in ways that reach far beyond anything most people appreciate, and understanding this shaping makes the transition of death and the dispersal of the microbiome after death more significant than it might otherwise seem. Consider what your microbiome is actually doing during your life. Your gut bacteria produce the majority of the serotonin in your body, not your brain, your gut. Serotonin is the neurotransmitter most associated with mood regulation, with the sense of well-being, with the capacity for emotional stability. Antidepressants that increase serotonin availability in the brain, selective serotonin reuptake inhibitors, are among the most prescribed medications in the world, and the majority of serotonin production in the body happens not in the brain, but in the gut, mediated by bacteria. Your gut bacteria produce precursors for dopamine, the neurotransmitter associated with motivation, reward, and pleasure. They produce gamma-aminobutyric acid, the primary inhibitory neurotransmitter of the central nervous system, which modulates anxiety, calm, and sleep. They produce short-chain fatty acids that cross the blood-brain barrier and directly affect neural function. They produce vitamins that the brain requires for normal function. They communicate with the brain through the vagus nerve, the long, wandering nerve that runs from the brainstem down through the chest and abdomen, connecting the central nervous system to the enteric nervous system of the gut. The vagus nerve carries information in both directions. The brain signals the gut, and the gut signals the brain. The gut contains more neurons than the spinal cord. It processes information about the body's internal chemical environment independently and continuously, and sends a constant stream of signals upward to the brainstem and higher structures. This gut-brain axis, as it is called, has been found to influence mood, anxiety, cognition, social behavior, and vulnerability to neurological disease in ways that researchers are still mapping. Disruptions in gut microbiome composition have been associated with depression, anxiety disorders, autism spectrum disorder, and Parkinson's disease, the last of which now appears in many cases to begin not in the brain, but in the enteric nervous system of the gut, moving upward through the vagus nerve over years before reaching the brain structures where the movement disorder becomes apparent. The implications of this for how we think about the self are significant. If the gut microbiome is producing neurotransmitters that influence how you feel, communicating with your brain in real time, and shaping the neural environment in which thought and emotion occur, then in some meaningful sense, your microbiome is part of you, not metaphorically, biochemically. The organisms living in your gut are participating in the production of your inner life. You are, in life, not a single organism. You are an ecology, a community, a negotiated coexistence between human cells and microbial cells that has been ongoing since before you were born. Since the moment you emerged from the birth canal and were colonized by your mother's microbiome, beginning the partnership that would continue for your entire life. When you die, that ecology does not cease to exist. It changes its purpose. The organisms that were participating in the production of your inner life, that were making your serotonin, your GABA, signaling your vagus nerve, training your immune system, become the organisms that dissolve you. The microbial community that was your interior partner becomes the agent of your transformation. And, as the body dissolves and its nutrients enter the surrounding environment, some of those microbial organisms disperse into the soil, into the water, into other organisms. The thanatomicrobiome succession continues until there is no substrate left to colonize, and then its members move on to other environments, carrying chemical signatures of the body they came from. What was once your internal ecology becomes, briefly, the ecology of decomposition, and then becomes part of the broader ecology of whatever place you die in. The community persists, dispersed, altered, distributed across multiple new hosts and environments. But the organisms are continuous. The chemistry is continuous. The information, in some molecular sense, persists. The history of near-death experience research is longer and more serious than most people know, and understanding it puts the current evidence in its proper context. The first systematic modern account of near-death experiences was Raymond Moody's 1975 book Life After Life, which collected accounts from people who had been resuscitated after clinical death, or who had come very close to death in other ways. Moody identified recurring elements across many different accounts from people who did not know each other and had not been exposed to each other's stories: the out-of-body experience, the dark tunnel, the encounter with a brilliant light, the meeting with deceased relatives, the life review, the boundary or threshold, the return. The book was a popular phenomenon and a scientific provocation. It invited serious researchers to ask whether these accounts contained information that standard neuroscientific frameworks could explain, or whether they presented genuine anomalies that required explanation beyond those frameworks. The response of mainstream medicine and neuroscience was largely dismissive. Hallucination, hypoxia-induced neurological noise, wish fulfillment, the brain playing movies from its archive as it shuts down. These explanations were plausible, broadly consistent with known neuroscience, and reassuring in their implication that nothing unusual was happening. But a number of researchers were not satisfied with dismissal and began systematic investigation. Kenneth Ring, a psychologist at the University of Connecticut, conducted the first systematic quantitative study of near-death experiences in 1980, collecting accounts from a large sample of survivors and finding consistent patterns across people of different ages, genders, educational backgrounds, and religious beliefs. His work established that near-death experiences were not isolated anomalies, but a consistent, recurring phenomenon with a recognizable structure. Michael Sabom, a cardiologist at Emory University, conducted a study in the early 1980s specifically designed to test the out-of-body claim. He interviewed cardiac arrest survivors who reported observing their own resuscitation from above, and compared their descriptions of the resuscitation procedure to what actually happened. He found that survivors who reported out-of-body experiences gave specific, accurate descriptions of resuscitation details that they could not have known through normal sensory means, details that matched medical records that were not visible from the perspective of a person lying on a resuscitation table, and that distinguished them from a control group who had not reported out-of-body experiences, and whose descriptions were generic and often inaccurate. This was a genuine empirical finding. It did not prove that consciousness had left the body, but it was not consistent with the simple explanation that the experiences were internally generated hallucinations with no connection to external events. Pim van Lommel, a Dutch cardiologist, conducted what became one of the most cited studies in the field, a prospective study published in The Lancet in 2001, tracking 344 cardiac arrest survivors across 10 Dutch hospitals. 62 of them, about 18%, reported near-death experiences. The experiences were not related to the duration of cardiac arrest, to the specific type of cardiac intervention, or to the level of anxiety or fear at the time of the arrest. They were not related to medication given during resuscitation. The patients who reported near-death experiences were not distinguishable before the event from those who did not report them in terms of personality, prior beliefs, age, gender, or other measured factors. Van Lommel's analysis led him to a conclusion that most of his colleagues found uncomfortable. The conventional assumption that near-death experiences are produced by a dying brain generating random hallucinations does not adequately fit the data. The experiences are too structured, too consistent, too independent of the factors that influence ordinary hallucination, and in at least some cases include verifiable information that the person could not have obtained through normal means. This does not prove that consciousness survives death. Van Lommel himself was careful to say he was not claiming that, but it does mean that the experiences require a more sophisticated explanatory account than dismissal as neurological noise provides. The tunnel experience, the sensation of moving through a dark passage toward a bright light that features so prominently in near-death accounts, has generated considerable neuroscientific speculation. Several explanations have been proposed. One is retinal ischemia. As blood flow to the eye fails, the retina may lose function at the edges first, because the peripheral retina has higher metabolic demand, creating the visual experience of a central bright area surrounded by darkness. Another explanation involves the retinotopic organization of the visual cortex, which may, under certain conditions of oxygen deprivation, generate spontaneous activation patterns experienced as light or tunnel-like structures. A third explanation, more speculative but genuinely proposed in the scientific literature, involves the activation of gamma-aminobutyric acid receptors in the visual system under conditions of extreme stress, which could produce cascades of neural activation experienced as tunnel-like visual phenomena. None of these explanations has been definitively confirmed or definitively ruled out. Each is consistent with some aspects of tunnel reports and inconsistent with others. The experience of moving toward the light, as opposed to simply perceiving light, is harder to account for with purely visual mechanisms. The sense of being drawn toward the light, the quality of intention and destination that many survivors describe, seems to involve more than a visual phenomenon. What is striking about the tunnel report is its cross-cultural consistency. It appears in ancient Egyptian accounts of the afterlife journey, in Tibetan Buddhist descriptions of the dying process, in Native American death traditions, in the accounts of contemporary cardiac arrest survivors in Europe, North America, and Asia who have had no exposure to these traditions. Something about the structure of human dying, in at least some cases, produces this particular phenomenology. Whether that phenomenology reflects the biology of a dying brain, or something about the relationship between consciousness and the physical world that we do not yet understand, is the question the data cannot yet answer. There is a phenomenon at the end of life that hospice workers and palliative care physicians observe regularly, and that is rarely discussed in scientific literature, but deserves mention: the deathbed vision. This is distinct from terminal lucidity, though related to it. In the days before death, dying patients, even those who are not cognitively impaired, who are fully conscious and communicative, frequently report seeing or sensing the presence of deceased relatives or other figures in the room. They speak to them or of them with apparent specificity. They describe being met, being accompanied, being assured. They reach toward figures that others in the room cannot see. A nurse named Maggie Callanan, working in hospice, documented hundreds of these cases in her 1992 book, Final Gifts, and described them as qualitatively distinct from the confusion of delirium, more structured, more consistent, more purposive. The dying person is not simply confused. They are describing something specific, something that appears to carry meaning for them, and the encounter often appears to reduce fear and distress. Whether these experiences reflect hallucinations generated by a dying brain, or something about the relationship between consciousness and death that our current framework does not account for, is unknown. What is documented is that they are common, that they are reported consistently across cultures and traditions, and that they have a characteristic structure. The figure is almost always a deceased relative or friend, sometimes a religious figure, and they are almost always experienced as coming to guide the dying person through the transition. Studies of hospice patients have found that between 50 and 60% of dying patients report such experiences in the final weeks and days of life. Among patients who are coherent and able to communicate, the percentage is higher. These are not rare edge cases. They are a regular feature of the dying experience, reported by the people living through it, with a consistency and specificity that simple dismissal does not adequately address. Whether these experiences are meaningful, whether they reflect anything about what death actually is, or what happens in it, is the question that neither the hospice literature nor the neuroscience literature has been able to answer. But the existence of these experiences, consistent and common, and shaped in ways that would be unlikely if they were simply random neurological noise, adds another layer to the picture science is slowly building. I want to say something directly about what I think the scientific evidence, taken as a whole, justifies believing. The evidence does not justify believing that consciousness survives death. The evidence does not justify that conclusion. The data from gamma waves in dying brains, from near-death experience reports, from terminal lucidity episodes, from deathbed visions, none of it rises to the level of proof of consciousness after death. The researchers themselves acknowledge this. The honest position is that the question is open, but the evidence also does not justify the confident dismissal that materialist orthodoxy sometimes offers. The claim that we know consciousness ends at death, cleanly, immediately, with certainty, is not supported by the evidence either. It is a prior commitment dressed up as a conclusion. What the evidence does justify is this: the dying brain is not simply shutting down. In at least some deaths, it is doing something unexpected and complex in its final moments. The experiences reported by people who have been resuscitated are not easily explained as random neurological noise. Terminal lucidity is a real phenomenon with no current mechanistic explanation. The hard problem of consciousness has not been solved, and the assumption that consciousness is simply what brains do may be less secure than the dominant scientific culture suggests. The appropriate response to all of this is continued investigation, intellectual honesty about uncertainty, and resistance to the temptation to decide the question before the evidence is in. The scientists who are doing this best, Parnia, Borjigin, Zemmar, Batthyány, Mashor, Koch, are holding the uncertainty carefully and continuing to gather data. They are not claiming more than the evidence supports. They are not dismissing more than the evidence warrants. They are doing what science is supposed to do when it encounters genuinely difficult questions. That is a good model to follow, not just in science, in life. Let me describe, in as much detail as the science allows, what the passage through dying may be like for the person experiencing it, not as certainty. As the most honest synthesis of what the data suggests, the heart stops. Within 15 to 20 seconds, the sense of being present in the world begins to fade. Whether this fading is experienced as a fading, or whether the last moment of awareness is simply the last moment of awareness and then there is nothing, is not known. The timeline is too fast for introspection to operate in any meaningful way. In some cases, the cases that produce the neural patterns we have been examining, something different happens. The brain, instead of fading into silence, surges. The temporal parietal junction lights up with gamma oscillations. The regions associated with memory retrieval and autobiographical continuity become more active than they were in normal waking life. If those gamma oscillations are accompanied by experience, if there is something it is like to be that brain in those moments, then based on what cardiac arrest survivors who were resuscitated have described, the experience might have these qualities: a sense of expanding clarity rather than dimming confusion, a sense of separation from the physical body, of observing the room from outside or above one's own physical position, an encounter with scenes from one's own life, not as sequential memory, but as simultaneous, panoramic, multi-perspective re-experience, a quality of moral completeness, of understanding, from multiple vantage points simultaneously, what one's life and actions meant to others. In some cases, a sense of moving toward a light or into a space of expansive peace. In some cases, an encounter with figures that feel like presences of deceased relatives or others. And in all reported cases, an encounter with some sense of boundary, of threshold, beyond which the person does not, in these accounts, go, because the person who would later report the experience returned. What is beyond that threshold is something no returned survivor has described. Whether it is continued experience, or silence, or something that has no analogue in normal human cognition and therefore cannot be reported, is unknown. The returning survivors describe the boundary as real. They describe something on both sides of it. Whether there is anything on the far side in the case of those who do not return, whether the threshold represents the end of experience or some other kind of transition, is the question the science cannot answer, and may never answer with certainty. But the threshold itself, in the accounts of those who returned from its edge, was not the end of experience. It was a location in experience, and that at least is interesting. The question of what death means, not biologically, but existentially, has occupied human beings since before recorded history. Every culture that has ever existed has had a practice around the dead. Every culture has had stories about what happens to the person who dies. The burial practices of Neanderthals, dating back at least 100,000 years, suggest that even our evolutionary relatives had rituals around death, may have believed in something that required tending to, some continuation or transition that deserved attention and ceremony. The universality of this is itself data of a kind, though its interpretation is contested. It could reflect the universal human inability to accept the finality of death, the hardwiring of social brains to continue relating to individuals even after those individuals are gone. It could reflect a universal intuition refined by evolution that consciousness is not simply a property of a body, but something that has a different and more fundamental relationship to the world. The science does not resolve this, but the science does add a layer to it that was not there a generation ago. The dying brain surges. The gamma waves appear. The life review has a neural correlate. Terminal lucidity exists and has no explanation. The zombie genes fire. The microbiome disperses. The atoms return. All of this is what we know now that William Bass did not know when he stood over that Confederate-era skeleton in 1977 and realized he needed better data. All of this is what has been built, piece by piece, in the labs and the hospitals and the body farms and the EEG monitoring rooms and the hospice wards in the decades since. The picture is more complex than anyone expected. The process is stranger and more active and more multidimensional than the simple language of death suggests. And the question of what experience, if any, accompanies that process, the question that cannot be answered from the outside and has not yet been answered from the inside, remains the most important open question in all of science. Not because the answer would change the biology. The biology is what it is, but because we are the organisms asking the question, and because what the answer is matters enormously to how we live with the knowledge that we will die. The courage to hold that question open, to resist both the consolation of a premature yes and the barrenness of a premature no, is part of what intellectual honesty demands. The instruments are measuring. The data is accumulating. The question is alive. Let the science settle into something quiet. The body you inhabit is a temporary arrangement of atoms that have been part of other things before and will be part of other things after. The community of organisms that makes you possible will dissolve and disperse, contributing to the soil and the air and the food web of the place where you die. The calcium in your bones will, eventually, be in the roots of things that grow. The carbon in your neurons will, eventually, be in the leaves of trees somewhere, in the bodies of animals, in the atmosphere itself. Whether the awareness that makes all this knowing possible, the awareness that right now is taking in this strange and layered account of what death is, Whether that awareness has any form of persistence beyond the biological cascade of dying is not known. The gamma waves are being measured, the terminal lucidity episodes are being documented, the thanatomicrobiome is being sequenced. The consciousness researchers are building finer and finer instruments to measure something that may or may not be measurable with instruments. But the question is being taken seriously, by serious people, with serious methods. And that is different from nothing. There is a particular kind of peace that comes, sometimes, from sitting at the edge of genuine uncertainty rather than false certainty in either direction. From saying, I do not know, and the not knowing is part of what makes this worth attending to. The great questions earn their status, not by having answers, but by remaining alive across every generation that encounters them. This one, what happens, if anything, after death, to the thing that was aware, is the oldest and the most persistent and the most personal. It will keep. The questions will still be there in the morning, and in all the mornings after, carrying us forward into whatever comes. Now let me take us somewhere that most accounts of this topic never go. The body as ecosystem, and why that framing changes everything about how we understand what death is. When William Bass founded the Body Farm and began documenting the decomposition of human remains, he was, without necessarily framing it this way, creating the conditions for a revolution in how forensic science understood the dead body. Before the Body Farm, the body was treated as an object, as evidence, as a puzzle to be read, as a static thing to be examined. After decades of Body Farm research, the scientific understanding shifted. The body is not an object. The body is a process, and after death, it is an ecosystem. Think about what happens in those first days after death, described now not in terms of forensic timelines, but in terms of ecology. The body is an enormous, concentrated source of chemical energy and biological nutrients in a localized space. Nothing in the surrounding environment even approaches its density of organic compounds, its concentrations of nitrogen and phosphorus and carbon and calcium. From the perspective of the organisms that can exploit it, bacteria, fungi, insects, scavenging animals, the arrival of a decomposing body in a local environment is like the sudden appearance of an extraordinarily rich island in an otherwise sparse sea. The ecological succession that follows is, from a biological perspective, a colonization event. The first colonizers are the bacteria already present in the body, now escaping their normal compartments. The early aerial colonizers are blowflies, attracted by volatile compounds from distances that can exceed a kilometer under favorable conditions. The blowflies are followed by secondary colonizers, beetles, ants, wasps, that exploit the opportunities created by the blowfly larvae. Later colonizers arrive as the conditions change, as the body dries, as the insect population shifts, as the chemical profile of the decomposing tissue changes. Meanwhile, underneath the body, the soil ecology is being transformed. The fluids draining from the body, rich in ammonia, in amino acids, in dissolved organic carbon, create a localized zone of intense microbial activity in the soil immediately below. Nitrogen-fixing bacteria increase in density. Fungi colonize the area. The network of fungal mycelium, which normally connects trees and plants across a forest or grassland through what ecologists now call the wood wide web, extends into the enriched zone. Studies at body farms have found that the area beneath and around a decomposing body is measurably more productive, biologically, than surrounding areas for years after the decomposition is complete. More microbial biomass, more plant growth, more insect activity, more soil nitrogen. The death of one organism creates a zone of enhanced life for many others. This is not, in the ecological framing, a tragedy. It is the way ecosystems are maintained. Large organisms concentrate energy and nutrients from diffuse sources, from sunlight and soil and air and food, and when they die, they release that concentrated energy and those nutrients back to the ecosystem in a highly available form. The larger and longer lived the organism, the more significant the nutrient pulse at death. A whale falls to the ocean floor and creates an ecosystem, what oceanographers call a whale fall, that supports hundreds of species for decades. A large mammal dies on land and fertilizes the soil for years. A human being, laying at the end of a life during which they consumed and concentrated resources from across a wide landscape, returns those resources in a concentrated pulse that benefits the local ecosystem measurably. This is what you are, from the perspective of the ecosystem: a temporary concentrator and accumulator of resources that will eventually be released. A node in the network that processes energy and matter through a period of years or decades, and then releases what it has concentrated. This framing is not cold, it is not reductive, it is accurate, and it carries a kind of dignity that the language of mere ending does not. The question of what death feels like, from the inside in the final moments, has been approached by science from an unexpected angle in recent years, through the research on psychedelic experiences. This connection might seem strange at first. Psychedelics are not death. People who take psychedelics come back. But the structural similarities between high-dose psychedelic experiences and near-death experiences have been noted in the scientific literature and are now being studied systematically. Roland Griffiths and his colleagues at Johns Hopkins University, whose research on psilocybin has been among the most important in the resurgence of clinical psychedelic research, have documented that high-dose psilocybin experiences often include what participants describe as ego dissolution, the temporary loss of the sense of being a bounded individual self, the dissolution of the ordinary boundary between self and world. Participants describe feeling merged with a larger reality. They describe encountering something that feels like the fundamental nature of consciousness itself, stripped of its usual personal content. They describe dying in experiential terms, and returning from it, an experience that a significant proportion of participants describe as among the most meaningful of their lives. The structural overlap between this and what cardiac arrest survivors describe is notable. Both involve the dissolution of ordinary self-boundaries. Both involve a quality of expanded awareness. Both involve encounters with what feels like a more fundamental level of reality than ordinary experience. Both tend to leave participants permanently changed, in ways that psychological assessments confirm are real and lasting. Reduced fear of death, increased sense of meaning and connection, increased compassion. A 2018 study in Frontiers in Psychology found that people who had undergone high-dose psilocybin experiences described the phenomenology as structurally similar to near-death experiences, with significant overlap in specific features: vivid imagery, feelings of transcendence, timelessness, unparalleled emotional insight, and a quality of seeming more real than ordinary reality. What this overlap suggests is not that psychedelic experiences and near-death experiences are identical. It suggests that they may be accessing similar biological mechanisms that exist in the brain and that are activated through very different routes by both extreme neurological stress and by certain chemical interventions. Whether those mechanisms are the brain generating compelling illusions, or whether they are revealing something about the nature of consciousness that ordinary waking experience conceals, is the question. But the consistency of the phenomenology across two very different circumstances, cardiac arrest and psychedelic administration, argues that whatever is being accessed is not random. There is structure here. There is a pattern. Something is being encountered repeatedly, through different doorways, and it is being described in consistent terms. The history of how human beings have understood and related to death is itself a kind of science, anthropological, archaeological, historical, and it adds context to the biological and neuroscientific picture. Every culture in human history has had rituals around the dead, which means that the transition of death has never been understood by any human community in any era as simply the cessation of a biological process. It has always been understood as an event requiring attention, care, ceremony, and some form of engagement with whatever the person has become after death. The ancient Egyptians devoted enormous resources to the dead, to the construction of tombs, to the preservation of bodies, to the preparation of supplies for the afterlife journey. The sophistication of Egyptian funerary culture, which spanned 3,000 years, reflected a specific and detailed theology of what happened to the person after death. A journey through a series of trials and assessments, a weighing of the heart against a feather, a negotiated arrival at a state of continued existence. The Book of the Dead, which accompanied wealthy Egyptians into the grave, was essentially a guidebook for navigating that journey, a text for the recently dead, providing instructions for the passages ahead. Whether the ancient Egyptians were right about any of this is beyond the scope of what science can say. But what their elaborate funerary culture reflects is an understanding that death requires navigation, that the person does not simply end, but goes somewhere, and that preparation for that going is an important responsibility of the living toward the dead. The Tibetan Buddhist tradition offers the most detailed and systematic account within any spiritual tradition of the dying process itself. What happens in the moments and hours and days after death, from the perspective of the dying person. The Bardo Fodol, known in the West as the Tibetan Book of the Dead, describes the dissolution of the elements that constitute the person: earth dissolving into water, water dissolving into fire, fire dissolving into air, air dissolving into consciousness, as a sequence of experiences that the dying person can navigate with understanding or without it. It describes a series of clear lights, increasing clarity, followed by visions, followed by further transitions, that parallel, in structure, if not in cultural content, the stages described in modern near-death experience accounts. The Tibetan tradition was not based on speculation. It was based on observation and practice, on the reported experiences of meditators who had trained extensively in dying-like states, on the accounts of those who had been close to death and returned, on the observations of practitioners present at deathbeds. It was a systematic attempt to understand and prepare for a process that was understood as the most important event of a human life. Whatever its metaphysical accuracy, it is a remarkable document of human observation of the dying process across many centuries, and the structural overlaps between its account and what modern neuroscience is now documenting are striking enough to have attracted the attention of serious researchers, including those at Johns Hopkins, whose psychedelic research explores states of consciousness that have traditionally been the domain of contemplative traditions. The question of what happens to the self after death is inseparable from the question of what the self is. If the self is simply the pattern of neural activity in a particular brain, if I am the pattern and not the substrate, not any particular collection of atoms but the organization those atoms are temporarily in, then what happens to the pattern when the substrate disperses? One interpretation is that patterns require substrates, and when the substrate disperses, the pattern simply ends. There is no pattern without matter arranged in a particular way, and when the arrangement fails, the pattern fails too. This is the most parsimonious explanation, compatible with what is known about how brains work, and it is the default position of most neuroscientists. Another interpretation is that patterns can exist at levels that are not fully determined by any particular substrate. In information theory, the concept of a pattern being substrate independent is well established. The same information can be encoded in different physical media, and the information is not identical to any particular physical encoding. Whether consciousness is a pattern in this sense, whether it can in principle be substrate independent, is one of the most contested questions in philosophy of mind. A third interpretation, associated with certain formulations of integrated information theory and with certain panpsychist views, holds that consciousness is not a pattern at all in the ordinary sense, not something produced by information processing, but a fundamental feature of physical reality that certain information processing systems instantiate or access. On this view, asking what happens to consciousness when the brain dies is like asking what happens to mass when a massive object is destroyed. The answer is that mass is not a property of any particular configuration of matter, but a feature of matter itself, and it persists in the particles that remain, even after the object is gone. None of these interpretations is proven. The hard problem of consciousness, why there is something it is like to be a brain processing information, remains unsolved. Which means that none of our theories about the relationship between brain and consciousness are on secure enough footing to confidently predict what happens to consciousness at death. What I find compelling, given everything the evidence suggests, is that the confident assertion of the first interpretation, consciousness simply ends at death, cleanly and completely, is not justified by what we know. It is the most parsimonious explanation, the one that requires the fewest additional assumptions. But parsimony is a heuristic, not a proof. And the evidence from dying brains, from cardiac arrest survivors, from terminal lucidity, from the hard problem itself, suggests that the relationship between consciousness and the brain is complex enough that the simple assertion of ending may be oversimplifying something deeply strange. There is a final dimension to all of this that I want to name before we close. The research we have been discussing was done by human beings who are themselves going to die. The forensic anthropologists at the body farms, the neuroscientists placing EEG monitors next to dying patients, the hospice workers documenting deathbed visions, the palliative care physicians who have sat at hundreds of bedsides and watched hundreds of people make the transition. This is not a feature of death research that applies to any other scientific field. The physicist studying black holes is not going to become a black hole. The ecologist studying rainforests is not going to become a rainforest. But the scientists studying death are studying what will happen to them. This proximity, this personal stake in the research, gives death science a quality that other sciences do not have. A quality of urgency and intimacy and, when it is done well, of extraordinary honesty. Batthyány has said, publicly, that decades of studying what happens to consciousness during cardiac arrest has changed how he understands what death is, and that the change has not made him more afraid of it. The data does not show a brain simply shutting down. The data shows something more complex, more interesting, more surprising than that. Whatever is happening in those final gamma surges, whatever the Aware 2 survivors were experiencing during their resuscitation, whatever terminal lucidity is, these phenomena are not consistent with the picture of death as simply the end. They are consistent with the picture of death as a transition, the nature of which we do not yet fully understand. Alexander Batthyány, who has spent 20 years collecting accounts of terminal lucidity, describes the phenomenon as the most challenging and the most important thing he has studied, not because it proves anything about the afterlife, but because it reveals that our model of the relationship between person and brain is incomplete in ways that have profound implications for how we think about consciousness, about dementia, and about death. Christof Koch, who lost his bet about the neural correlates of consciousness and has been engaged with the hardest version of the hard problem for 30 years, has arrived at the position that consciousness may be more fundamental to the structure of reality than his earlier materialist commitments allowed him to consider. He remains a rigorous scientist. He does not claim more than the evidence supports, but he is willing, in a way that reflects genuine intellectual courage, to acknowledge that the question is bigger and stranger than the comfortable dominant view suggests. These researchers have looked at death more closely than almost anyone, and none of them have arrived at simple certainty. They have arrived at complexity, at genuine mystery, at the kind of wonder that comes from knowing enough to understand how much is not known. That seems like the right place to arrive. Now the long account is done. The cascade has been described, from the moment the heart stops, through the cooling and stiffening and dissolution and return of atoms to the cycle. The brain's final surge has been described, and the experiences of those who have come back from its edge. The terminal lucidity has been examined, and the zombie genes, and the phanotomicrobiome, and the organ donation clocks, and the history of how death has been defined and redefined. What remains is a quality of attention, the quality that comes from having sat with hard material honestly, without flinching away from it, and without reaching for false comfort. Death is real. It happens to everyone. It is more complex, and more active, and more strange than the simple word suggests. It ends the particular arrangement of atoms and biology and information that constitutes a person. It may or may not end whatever that arrangement experienced as awareness. The question is open, genuinely and scientifically open, and it will be open for some time yet. But what is not open is that the atoms return. The chemistry continues. The ecology of the place where you die is enriched by what you were. The organisms that were part of you disperse into the world. Something of the concentration and order that was a life goes back into the system that made it possible, and whether that is all there is, or whether it is part of something larger, is what the instruments are still measuring. Let the questions carry you gently into sleep. The night holds all of it. There is something worth saying about the act of waiting. In palliative care, the period immediately before death, sometimes hours, sometimes days, is known by practitioners as the active dying phase. The breathing changes. Long pauses appear between inhalations. The hands and feet cool. The skin around the nose and mouth takes on a particular blue-grey cast that nurses and hospice workers learn to recognize immediately. Consciousness comes and goes. There are periods of stillness so deep that those keeping vigil find themselves leaning in to watch for the rise and fall of the chest. Hospice workers describe something else that happens during this phase. A change in the quality of the room. A quality of attention that arises in those present. A slowing, a quieting, a sense that something significant is happening that cannot be hurried or managed. Even people who have sat at many bedsides describe this. It is not grief exactly, not yet. It is something more like witness. The dying person, in those final hours, is often reported to be communicating, eyes moving, lips moving, expressions of recognition on a face that by every clinical measure should not be capable of recognition. The electroencephalogram that Zenma recorded showed gamma activity. What Zenma could not record was what it felt like from inside, whether anything was felt at all, whether the movements and expressions were the residue of old neural patterns firing without awareness, or whether they were the sign of something that was still, in some sense, there. This ambiguity is not a failure of science. It is the current location of the frontier. The instruments that could resolve it do not yet exist. The questions that would need to be answered to resolve it have not yet been precisely enough formulated. What science has done is bring us close enough to the edge of this question to see that it is a genuine question, not a theological assertion, not a wish, but a real uncertainty with real evidence on multiple sides. The biology of grief is less studied than the biology of death, but it is part of the same system. When someone who has been close to a dying person, who has sat with them, who has held their hand in the active dying phase, who has witnessed the final breath, leaves the bedside afterward, they carry a neurological imprint of what they have witnessed. The stress hormones remain elevated for weeks. The immune system is measurably disrupted. Sleep architecture changes. Appetite changes. The brain regions associated with social bonding, the same regions that process the expectation of another person's presence, continue to send signals, expecting a response that no longer comes. This is what grief looks like in biological terms. The body continues to reach for a connection that the mind knows is gone. The neural patterns that were built around the presence of the person do not dissolve quickly. They persist, fire without their expected answer, and the mismatch between expectation and reality is experienced as loss. Some researchers in the neuroscience of grief have argued that this persistence is not a flaw in the system but a feature, that the biological cost of grief reflects the depth of the investment that social bonds represent, and that a species capable of investing so deeply in others would necessarily pay a high cost when those bonds are severed. The capacity for grief is the shadow side of the capacity for love, and neither can be reduced without reducing both. What this means, in the context of a conversation about what death is that death does not end the relationship between the dead and the living. It changes it, transforms it, reroutes it through memory and the persisting neural patterns of those who remain. The person who has died continues to exist, in a real neurological sense, in the brains of those who knew them, continues to shape responses and expectations and the structure of daily life for as long as those people live. This is not the same as survival, but it is not nothing. The last thing I want to say is about time. Death is the end of a person's time, the end of their particular experience of duration, of sequence, of before and after. What happens after death, whatever it is not inside that person's time. It is outside it, in the time of the world, in the time of atoms and ecosystems and the geological record and the stars. What strikes me at the end of this long account is how different those two timescales are, and how difficult it is to hold them both at once. From the inside, a human life is enormous, rich with detail, filled with irreplaceable particular moments, dense with relationships and choices and discoveries. From the outside, from the timescale of the atoms that compose a person, which have existed for billions of years and will continue to exist for billions more, a human life is brief in a way that is difficult to comprehend. A flash, a temporary arrangement, a moment of complexity in a very long history of complexity. Both of these perspectives are accurate. The inside perspective is the one we live in. The outside perspective is the one that is, in a literal sense, more durable. The atoms will outlast everything that can be said about what it felt like to be us. And somewhere between those two perspectives, between the irreplaceable richness of a single human life and the vast indifferent continuity of matter cycling through forms, is the actual mystery of what death is, and whether the inside perspective is something that ends with the ending of the arrangement, or whether it is something that the science of the 21st century is only beginning to have the tools to approach. Let that question accompany you into the rest that follows. Sleep is a kind of boundary too, a temporary dissolution of the structured waking self into something quieter, more dispersed, less defined. You cross it every night without ceremony, and you return each morning without fully knowing where you were. The gamma waves that fired in those 4 monitored patients as their hearts stopped were real. The EEG recorded them. The temporal parietal junction, the region associated with the sense of being a self located in a body, surged with activity at the precise moment when the body it had organized was ceasing to function. Whatever that surge was, a last computation, a release, an artifact of dying tissue, or something that our current models of consciousness cannot yet account for, it happened. The instruments caught it, and the researchers who caught it have not stopped thinking about what it means. That is where the science currently stands, at the edge of something it measured but cannot yet explain, which is, when you consider it honestly, exactly where science should want to be.